when tiny particles called exosomes, which shuttle signals between cells, are defective in diabetic patients, they can drive inflammation and impair healing of wounds, according to a new study published in the journal Nano Today by University of Pittsburgh and UPMC researchers. In chronic wounds of diabetes patients, these faulty exosomes can't deliver vital information to cells that promote wound healing, the researchers found. These insights open the door to new exosome-focused therapies to promote healing of chronic wounds. In patients with diabetes, wound healing is impaired because of excess inflammation. Left untreated, these non-healing or chronic wounds can lead to limb amputations. More than 100,000 diabetes-related amputations occur in the US each year, but by understanding more about wound healing and developing new therapies, the researchers aim to bring down this number. Using negative pressure bandages that gently vacuum wounds to stimulate healing, the research team collected wound fluid from chronic wounds of 22 diabetic and 15 non-diabetic patients. The researchers isolated and analyzed exosomes produced by skin cells called keratinocytes. After these particles are packed with cargo, including RNA, lipids, and proteins, they are released from the cell and taken up by macrophages, immune cells that coordinate wound healing. If signals contained within exosomes are correct, the macrophage knows how to resolve inflammation in the wound. In diabetes, crosstalk between keratinocytes and macrophages is compromised. So, macrophages keep driving inflammation and the wound can't heal. Diabetic exosomes, which the researchers dubbed dia-exosomes, had different contents of RNA, lipids, and proteins than those from non-diabetics, suggesting that the cargo packing process is altered in diabetes. Diabetes also compromises release and uptake of exosomes in wounds, the research team found. The number of dia-exosomes in wound fluid from diabetic patients was much lower than exosomes in non-diabetics, and macrophages took up far fewer exosomes than dia-exosomes. When the researchers incubated non-diabetic macrophages with exosomes, the macrophages produced compounds that signify resolution of inflammation, indicating that they had received the exosomes message and responded correctly to initiate wound healing. But when they repeated this experiment with dia-exosomes, the macrophages produced pro-inflammatory compounds common in diabetic patients with chronic wounds. Dia-exosomes drive deviation from the healing cascade so that resolution of inflammation is compromised. And this isn't just limited to wounds. Because exosomes are responsible for many functions in the body, dia-exosomes could play a role in other diabetic complications. This study opens a new line of thinking. The researchers are now investigating how they could target dia-exosomes to improve wound healing in diabetics. One avenue is to develop therapeutics to undo chemical modifications that occur in dia-exosomes. Alternatively, they could isolate exosomes from diabetic patients and load them with missing signals before infusing them back into the wound tissue.